Hey everyone, I hope you can hear me and see me, and also the slide as well.
We can see everything.
Awesome. Okay, thanks everyone for coming in person as well. It's my great honor to be here actually to give a seminar at Rutgers. I'm particularly excited because a lot of work from here and from Dina's lab especially are underlying my PhD work, as you'll see a lot in the slide. So let me briefly introduce myself. I'm originally from Tokyo, Japan. I was born and raised there. And I finished my undergrad at University of Tokyo, studying the evolution of social morads. And then I switched gear to insects, which everyone here loves, to study berry beetle in Asia, working with Dustin Rubenstein at Columbia University. After my master's, I moved to Princeton to do my PhD with Lindy McBride, working on the genomics of mosquitoes and their behavior, as you'll hear today. Outside work, I love soccer and ramen. And I heard that New Brunswick has one of the best Japanese ramens in the United States. I think it was like a Nagomi ramen or something. I don't know if you guys know, but I really need to try that out after this seminar. So I'm really excited about that as well. Okay, so that's enough for my introduction, and I'll just jump into the talk. So as we populate the world, we've been changing every corner of the earth dramatically. And since tens of thousands of years ago, our agriculture and farming have engineered the landscape significantly, as you can see on the top pictures. And as we all know, the impact has been only accelerating, particularly in the past hundreds of years. We've created many complex and beautiful urban megalopolis around the globe, from Cairo to New York City to Tokyo. This global engineering of the earth, both directly and indirectly, impacted all lives on the earth and led to the unprecedented era of the evolution called the Anthropocene. Many mammals like foxes have adapted to urban environments, and you can even see them bossing around in Princeton. And if you look down, you'll find some plants popping out of the concrete, surviving well in that really harsh environment. And pepper moss, as you all know, color evolved to adapt to contaminated bark on the tree. And really amazingly, they kind of evolved back to white when the air got cleaner. And even you can see the New York City rats adapting to eat the pizza, officially claiming the New Yorker position. So these are just a handful example of animals and plants that have adapted and evolved in the Anthropocene. And here's a map of more example, and just to show you the breadth of the life adapting to human habitats. And as you can imagine, the list is only growing every year. Among the most fascinating examples, though, are mosquitoes adapting to human habitats. And I'm sure that this crowd actually can agree with me. I don't have to really convince you guys. But as you know, there are about 3,000 species of mosquitoes in the world, but the only handful of species that specifically adapted to humans or human environments are the ones that make millions of people sick and kill hundreds of thousands of people every year by vectoring horrifying pathogens like malaria, dengue, Zika. So just to show a couple examples, they evolved an array of ecology and behavioral traits adapted to human environments. And on the left, Anopheles gambi, their larvae are living specifically in agricultural ditches, and the adults are specialized in biting humans. And on the right, Aedes aegypti, which our lab primarily studies, also evolved to breed in artificial water containers and also specialized in feeding on humans as well. So these are the species that you hear the most. Those species that vector horrifying diseases primarily in Africa or developing countries as well. But today, I like to tell you about another disease vector. Oh, sorry. Mosquito. I think you can turn down the. Sorry. Yeah, today I'd like to tell you about another disease vector mosquito that you may know well, found across the temperate region that has received much less attention. To do so, I'd like to take you back in time first with me to a cold winter in London, 1940. In the winter of 1940-1941, the Second World War was raging, and Londoners faced a sustained 8 months Nazi bombing campaign known as the Culex. During one particularly intense period from September to November, bombs were dropped for 57 nights in a row. Many people who had lost their homes or simply wanted a more secure place to sleep looked for a nightly refuge in the city's underground subway system, or the tube. Oh, sorry. In the tens of thousands of men and women and even children slept shoulder to shoulder on station platforms, escalators, and sometimes in the subway tunnels themselves. The underground system provided literally life protection from bombs for them. However, the Londoners who took refuge there found themselves attacked by another enemy, mosquitoes. The name of the mosquito is Culex pipiens for molestus, also known as the London Underground mosquito. It turned out that the London Underground had also become a home to this urban ecotype of the widespread temperate mosquito. And this little creature is indeed the main character of my presentation today. Scientists have studied the populations and found that there are two behaviorally divergent forms within Culex pipiens mosquito. These pesky biters found in London Underground had adapted to life in man-made below-ground structures like subways or basements. They were active year-round, voraciously attacked humans, and could mate in confined spaces. Seeing such a mosquito perfectly adapted to man-made subterranean structures was surprising to the people at the time, because if you compare molestus to the more common above-ground form pipiens, it diaposes in winter. It mostly bites first, and actually they are known as man-ignoring form of Culex pipiens and requires a large space to swarm in to mate. Strikingly, despite the array of behavioral and physiological differences, the two forms are morphologically identical. So even vector biologists cannot tell them apart by just looking at them. When I started my PhD, I decided to develop this fascinating system into a model system of genomics of rapid urban evolution. And that's firstly because this system is just so interesting and bogus in its own right, but also because I was convinced that studying this system can help us tackle some of the fundamental questions about the evolution in the Anthropocene. So for my PhD thesis, I'm asking following specific questions. First, to understand the origin, status, and evolutionary history of the two forms of Culex pipiens, I'm asking whether the two forms are genetically distinct in the first place, and when and where the below-ground form originated. Second, taking advantage of naturally hybridizing populations of Culex pipiens, I am asking to what extent do the forms and subspecies hybridize, and how does the hybridization affects the ecology and behavior of those populations. Finally, I'm asking what are the genetic mechanisms underpinning the behavioral differences, which is going to be my fourth chapter. And overarching goals are to inform evolutionary biologists, public health experts, and feeding our curiosity a little bit. For the interest of time today, I'm going to talk mostly about the origin story of the molestus, but feel free to ask later about other projects if you're interested. Okay, so let's start with the origin question. When and where did the below-ground form originate and evolve? So the major hypothesis about the origin of molestus is what we call the in situ hypothesis. It states that below-ground molestus populations evolved from above-ground populations in situ. And this suggests that the evolution happened within less than 100 years, because the very first station in the tube was built in 1860s, and the London population was found in 1940s. As you can imagine, the in situ hypothesis never really ceased to fascinate academic scholars and public audience alike. With some help of surging interest in urban evolutionary biology in recent years, many reviews in renowned journals like Science repeatedly cites the in situ hypothesis, hailing molestus as the best supported case consistent with the process of urban speciation. You might recognize some of the names here because they are like who's who in evolutionary biology. The public media, including BBC, New York Times, Guardian, also wrote about molestus being the unique mosquito that lives in the London underground. And I want to call your attention to Bill Nye specifically, because in my mind, he's like the kind of king of science communication for kids in the United States. And he says the evolution of underground mosquito happened over the span of approximately 100 years. Evolution, am I right? So in the beginning of my PhD, I remember reading all this and kind of thought to myself, is Bill right? And as everyone here would do, I started by looking for literature that unequivocally supports the in situ hypothesis. And turns out all the reviews citing molestus as the proof of concept of urban speciation has mainly relied on this one study published in 1999. The authors looked at both above and below ground populations in London and investigated allozyme loci, which is kind of an old way to look at genetic structure of population, and found that the above ground London populations were genetically distinct from the below ground London populations. So blue circle and red circle. Well, it seems true that they are genetically distinct and probably reproductively isolated in London, but it doesn't say much about the origin, when and where the below ground populations first evolved. Yet they claim that below ground population evolved in London underground from the above ground population. So at this point, I was kind of scratching my head and looked further into the literature. By gathering more studies, looking at local populations here and there, it became clear that the distribution of below ground populations is global, just like the distribution of above ground populations across the temperate. If all the below ground populations evolved from nearby above ground populations in the past couple hundreds of years, which the in situ hypothesis kind of predicts or implies, that would be awesome. But as evolutionary biologists, we know how unlikely that would be. And instead, this map made us wonder if the molestus populations actually evolved once in a different context, and these populations across the world are migrants. So at this point, I was like, Bill, it's not looking great for you. And we bumped into this study published in 2004, which had been largely ignored by the evolutionary biology community. Here, our very own Dina Fonseca at Rutgers and her colleagues investigated the relationships between both above and below ground populations in Europe using a handful of microsatellites. They found that the below ground molestus populations clustered with each other, suggesting that there was likely a single origin for all the below ground populations in the region. Also, they did not cluster with nearby above ground populations, suggesting that they were not likely derived from the nearby above ground populations. Furthermore, she found that the above ground individuals from the Middle East were closely related to other below ground populations. She posed the hypothesis then that Middle Eastern populations were somehow ancestral to the below ground populations in the north. But from this genetic data alone, it could be still that the molestus populations evolved in the northern city first and came to Middle East. Unfortunately, this study, again, has not gotten as much attention recently as the in situ hypothesis in the evolutionary biology community, probably because the data was not sufficient both in terms of the limited sampling effort and the sparseness of the genetic markers, although it was impressive at the time. Nevertheless, we were really motivated by the intriguing inconsistency between the existing data and the in situ hypothesis and decided to conduct a comprehensive meta-analysis and review of existing studies to see if we can generate a model that explains the pattern we see here. So in looking through almost a century of literature published between 1920 to 2020s, we looked for a standardized method to investigate the genetics of Culex pipiens populations, which we can then take to see the pattern across different regions. Luckily, there's one method called CQ11 assay, which looks at allelic variation near a microsatellite locus called CQ11, which again, Dina and her colleagues developed. It was developed based on ecologically well-defined pipiens and molluscus populations, such that one allele is fixed for pure reference molluscus populations, and the other allele was fixed in pure pipiens populations. And the advantage of the method is that you can literally look at the size of amplified fragments without sequencing them, as you can see in the gel picture, allowing for a cheap and quick way to diagnose the populations for forms. Remember, pipiens and molluscus are morphologically identical, so this method became really popular and widely used even now. The studies looking at CQ11 are often very local and fragmented, but we can synthesize data from these local studies to see if there is any emerging pattern. And here's what we see. Here, each pie represents a population, and the blue color indicates the proportion of individuals homozygous for pipiens allele at the CQ11 locus. Red indicates the proportion of individuals homozygous for the molluscus allele. And yellow indicates the proportion of heterozygous individuals. On the left, you have above ground populations, and on the right, you have below ground populations. First, you can see that in the northern populations, like in the UK or Sweden, above ground and below ground populations are more or less fixed for pipiens and molluscus alleles respectively, confirming the pattern for pure pipiens and molluscus. However, as one moves to the south, you see something different. Above ground populations seem to get more and more molluscus alleles, and below ground populations seem to get more and more pipiens alleles. And at the southern edge, like in Morocco here, the difference between above and below ground populations is much less clear. So I can summarize the latitudinal pattern of allele frequencies in these maps into a graph like this, where each circle represents above ground population and each triangle represents a below ground population. On the y-axis, now you have latitude, and on the x-axis, you have frequency of pipiens alleles. And the frequency of molluscus allele, by the way, is just 1 minus p. Here you can see clearly that in the north, above ground and below ground populations are really distinct genetically, while in the south, distinction seems to be blurred. At the southern edge, the above and below ground populations seem to be not really different and well mixed with respect to the CQ11 locus. I just want to quickly note that we also found a lot of ecological studies before 1980s and 1970s, which is, pre-genetic era. The southern populations show mixture of pipiens and molluscus features. So for example, the above ground populations in Portugal or Greece seem to be more mammal biting compared to the Swedish above ground pipiens populations. So with the existing data hinting at the surprisingly clear latitudinal gradient, we are now proposing a new model clarifying the genetic status of the two forms of Culex pipiens. Here on the left, I have colored above ground populations as background and depicted below ground populations as scattered circles. The main take-home message here is that there is a latitudinal gradient of pipiens and molluscus ancestry, both in above and below ground populations. The model also predicts that the dichotomy of above ground pipiens and below ground molluscus seen in the north breaks down in the south. Why, you may wonder? The main reason we can think of is the presence of the cold winter in the north. So imagine you are a molluscus in London, right? You are mainly breeding in the below ground, but you may happen to try to breed above ground in the winter and just fail because it's simply too cold outside. This keeps molluscus, who is breeding all year round, in the subterranean places where the temperature stays preferable around the year. But such selective pressure weakens as one moves to the south. So in the low latitude, such selection might not exist anymore, and the above and below ground populations distinction disappears. Thus, by synthesizing published genetic data, we started to see what was missed by the overly simplified narrative of above ground versus below ground populations dichotomy. But it did not cover the important Middle Eastern region because there has been no CQ11 studies published so far from that region.
Maybe just one request from the audience.
Yeah.
Use your cursor to point things in the map.
Yeah.
When you're trying to point out to specific things.
That-
Can you see your cursor? I'm not
That
seeing
makes
your
sense.
cursor. Oh, there you go.
Do people see my
Yeah,
cursor?
You can see your cursor.
Okay, great.
All right,
Okay.
I did it.
Thank you. Okay. So yeah, I just said that CQ11 data was not published from that important Middle Eastern region. To overcome that situation, we next turn to behavioral data, specifically host choice data based on blood meal analysis, for which you may be really familiar. Here, each pie represents, again, a population and the proportion of collected mosquitoes with blood. Red indicates the proportion of mosquitoes with mammal blood, including human blood. And blue indicates proportion of mosquitoes with bird blood, bird and mammal. These blood meal data can be used as a proxy of host preference for each population. And while we see a similar climb of north to south until Mediterranean region, although the correlation is much less clear compared to the genetic data, the Middle Eastern population looks something really different. They're all predominantly mammal biters, and in fact, they are largely human blood. This pattern suggests here, sorry. This pattern suggests that the Middle Eastern populations are behaviorally similar to the below-ground molestus populations in the north. Actually, in the vector biology field, the human-biting Culex pipiens in Middle East have been long known. The Culex pipiens populations in the Mid-East are known to vector filaria worms here that cause lymphatic filariasis, which was a huge problem in Egypt during 1930s to 1980s. This species of filaria worm can reproduce only within humans. Therefore, it requires human-biting mosquitoes to present and vector them around humans. In fact, the filaria worm and lymphatic filariasis, sorry, my pronunciation is not good, is almost absent in the other parts of Europe. So how old then are the human-biting populations in the Middle East? Are they derived from northern below-ground molestus, or they are older? There's some evidence that the human-biting populations have been around for a while in the Middle East. This is an artifact. This one is an artifact of an ancient Egyptian king, Mentuhotep II, 4,000 years ago, which famously had enlarged legs, a signature symptom caused by lymphatic filariasis. So these suggest that the main biting Culex pipiens in the Middle East are actually much older than the below-ground molestus in the north, and probably ancestral to them. But one question still remains: why would mosquitoes have evolved non-biting in the Middle East to begin with? If we look at the history of human cultural evolution, there are many reasons to believe that the evolution of human adapted mosquitoes might have happened in the Middle East on the order of thousands of years. The Middle East is precisely the region where the earliest agriculture was invented, which could have provided plenty of opportunities for mosquitoes to adapt to human environments. We can imagine that in these very arid regions, the agricultural water source might have been the only place for them to breed. And for the adult mosquitoes, human and some domesticated animals must have been the only host available at the time. These regions even had below-ground water system called kanat, which people used for irrigation purposes. And this kind of structure may have further helped mosquitoes to evolve the ability to reproduce in an underground confined spaces. Then it makes good sense to think that these pre-adapted populations in the Middle East have colonized the northern subterranean habitats, like the London Tube more recently, probably on the order of hundreds of years. So after all, Bill, it doesn't look like you're right. Sorry. But Bill may not be convinced yet fully because our model still relies mostly on the one locus study. And the geographic coverage of the data points are far from complete. So to really convince Bill and ourselves for the next step, I decided to test this model with comprehensive sampling and genomic data. And that's why we started PIPOP, the Culex pipiens population genomics project. The aim of this project is to sequence around 1,000 genomes from both above and below ground populations to understand the status, origin, and genomic basis of the Culex pipiens forms. We started this project back in 2018, and at the time we didn't have any connections within the Culex research community because the lab back then worked exclusively on Aedes aegypti. So Lydia and I decided to do our own version of the Blitz, which is the email Blitz. We created a flyer like this on the left side. So this is a PDF flyer, and bombarded with emails, literally every single Culex pipiens researcher, including Dina, for a collaboration. It's like door knocking in fundraising campaign, but it's virtual and looking for dead mosquitoes. So we also utilized the massive social capital that Linda had on Twitter, announcing people far wide in the internet world. Meanwhile, I got my hands dirty and collected mosquitoes locally in the Northeastern America. For the above ground collections, I brought this CDC light trap here to Princeton, New York City, Philly, Boston to collect above ground mosquitoes. And this is for like a general audience slide, you know, people who may not know CDC light trap, but I think in this audience it doesn't, it's not necessary. Below ground collections turned out to be more difficult because these human biting populations tend to be reported and immediately extirpated by local public health agencies. But with tons of help from a collaborator in University of Maryland, I went to the below ground floors of parking lots in DC to collect adult below ground mosquitoes using this really cool tool called Insectazuka to suck the adult mosquitoes. And sometimes I had the privilege to stick my heads into the hole with some warm water, what kind of water, I don't know, at the bottom to collect larvae. And it's actually very exciting to see these below ground molluscs in their natural habitat, quote unquote. So after 3 years of collections, here's what we got. Thankfully, we got a lot of enthusiasm about this project from Culex pipiens researchers all over the world, from Americas, Europe, Africa, Asia, and Australia. So far, we've got more than 12,000 samples from 90 groups coming from 5 different continents. One thing I want to emphasize here is that they're not only university researchers, but a lot of local public health workers collecting and counting mosquitoes every day. And they may not publish academic papers routinely, but I could feel their curiosity and enthusiasm about the mosquitoes and their evolution. So the interactions with them actually made me even more excited to answer the evolutionary questions of this species. We've sequenced more than 700 individuals so far, but for this part of my talk, I will first focus on the 355 individuals collected from Western Palearctic area. In other words, Europe and North African regions, which we have been talking about today. Okay, let's just jump right into the genomic analysis result. This is a PCA plot of the 355 sequenced individuals from the Europe and North African regions using millions of SNP data. Each point now represents one individual, and circles represent individuals from above ground sites, and triangles represent those from below ground sites. I hope the virtual people can also see. What was immediately clear to us was that on the left, we have below ground molluscous individuals, and on the right, you have kind of like a wide range of above ground individuals occupying the PC, almost like the full range of PC1, which we think indicates the pipiens to molluscus axis. So first, I can say from this plot that there seems to be some genetic continuity, not like discrete clusters exist among the individuals here. But let's take a step back and remember that our goal here is to ask if pipiens and molluscous ancestry correlate with latitude, as predicted by our model earlier. So our prediction would be this kind of V shape like this, where at high latitude, pure above ground and pure below ground individuals are two distinct clusters. And at low latitudes, however, individuals may be intermediate and well mixed. Okay, let's see what data say. Ta-da! This is indeed what we see. So in the nose, above ground pure pipiens and below ground pure molluscus are nicely distinct and are on the both sides of the PC wall. Farther south, the Mediterranean above ground individuals occupy a wide range of the PC1, starting from the pure pipiens-ish values to more molluscus-like above ground populations. This suggests that in this region, pipiens-like individuals and molluscus-like individuals coexist with extensive gene flow. And most excitingly, the above ground individuals from the southern edge of the distribution, or the Middle Eastern individuals, are very molluscus-like without pipiens individuals nearby, consistent with our model as well. So we are particularly excited to see this because we had almost no published genetic data from this region. Together, our individual genomic data confirm the predictions from the latitudinal gradient model, which we built earlier from more patchy and sparse genetic data. Okay, the prediction about the genetic status looks good. But what about the origin and evolutionary history of the forms? Now that we established the gradient model is likely true, next we wanted to use whole genomes to understand the origin and pathway of spread across the European molluscus populations. More specifically, we can start asking two key questions regarding the origin of molluscus. First, is the Egyptian population ancestral to other molluscus populations in the region? And second, when did pipiens and molluscus split in the tree? Key predictions from our model to answer those questions are, first, if Egyptian molluscus is indeed ancestral, then Egyptian molluscus would be basal in the tree. And if we think that the northern populations are indeed migrants from the south, then the Egyptian populations would be the most diverse populations when compared to more northern populations that are supposedly derived. Second, our model predicts that the molluscus and pipiens populations split on the order of thousands of years ago, not hundreds of years ago, as the in situ hypothesis would predict. Okay, let's dig into this, starting from a phylogenetic tree. Here's a population phylogeny for the European and North African populations with a lot of outgroup populations clasped on the top from Asia and Africa, for example. The European above ground pipiens populations, first branch of the tree, we should use this, which is distinct from the EU below ground populations here, although there is a variation within the tree. And the Middle Eastern populations, which are molluscus from Egypt and Israel regions, they are, at least in the tree, looking basal to the below ground ones, indicating that these populations are likely ancestral to the below ground molluscus populations. But the tree shape, it's not enough to say that these are ancestral to other molluscus populations. So we looked at the genetic diversity across the genome to confirm if they are actually more diverse than other derived populations. And this is the diversity across the genome, across the windows of the genome, to be more precise. And we found that the Egyptian molluscus is the most diverse among all the molluscus-like populations, suggesting that Egyptian is indeed the ancestral molluscus population. Furthermore, we also found a more general trend that southern populations of molluscus is more diverse than northern populations. That the northward expansion of molluscus, presumably via human kind of bringing or spreading with mosquitoes into the northern Europe, is likely true. Currently, we are working on a more explicit demographic modeling approach to nail down exactly which populations are likely the source of which populations downstream. Okay, so Now we established that Middle Eastern molestus is likely the ancestral molestus population. What about the question as to when exactly the pipiens and molestus split from each other? To know when pipiens and molestus exactly split, we inferred trajectories of population size through time using a method called pairwise sequential Markovian coalescent, or simply PSMC. Briefly, this method scans across the individual genome, in this case, a diploid genome, to collect independent coalescent trees across many loci, and infers population history that makes such distribution of coalescent trees possible. As an example result, here I'm showing you the population size histories of two groups of orangutan in orange and yellow. On the y-axis, you have the population size, and on the x-axis, you have time before present. So note that on the left, you have time closer to present, and on the right, time far in the past. You can see that two populations have shared population size history up until this point, and then kind of these two populations start to split in terms of demography. So we can infer that those two populations have split around 1+ million years ago. So I applied this method to our genome data of above-ground pipiens and below-ground molestus to see when exactly those above and below-ground individuals split in the north. So here in red, we have below-ground molestus, and in blue, we have above-ground pipiens. We do see shared history of pipiens and molestus until this point. Maybe there's a little bit of divergence around here too, but the two started to clearly diverge from here. The estimate indicates that the split time is around 10,000 years ago, which is far older than the London Underground subway system, to be sure. And instead, it nicely corresponds with the era of agricultural revolution in the Middle East. So in summary, I've been exploring the genetic status and origin of the enigmatic ecotypes of Culex pipiens mosquitoes. First, I conducted a thorough review and meta-analysis to synthesize existing published data, finding little evidence that supports the prevailing in situ hypothesis. Based on the emerging pattern from, second, using a global set of samples and whole genome sequencing, I tested the predictions from our model, finding that the data supported the latitudinal gradient model and suggested an older southern origin of the below-ground molestus. So what is next then? Well, there are a couple more exciting ongoing projects based on these results. First, and as an extension to our model in Europe and North Africa, we are now building a more precise distribution model of pipiens, molestus, and intermediate form across latitudes, like this. Using the model and future climatic predictions, we can now start to infer how the distribution shift in the near future. Remember, these ecotypes can't be separated by morphology, and yet the ecology is so different. So such genomics inferred modeling is particularly useful for both disease vector ecology and evolutionary biology communities in these regions, because maybe what you get and what you see under the microscope is not really going to be different in 20 years, but the behavior and ecology might be super different from what you see now. Second is what we call Project Going Underground. As I mentioned, the story really started with the London Underground subway system, and I really hope to get some contemporary samples there. But unfortunately, we couldn't get any. It may be that London Tube is now a lot cleaner than it was in 80 years ago. But we are now collaborating with people at the Wellcome Sanger Institute and the Natural History Museum in London to sequence molestus specimens from 1940s to 1980s, when people first found out about the pesky biters in London. So this is the specimen, actual specimen picture, and they look really intact. And yeah, I don't know, it's just amazing how we can even sequence them from their genetic material. So stay tuned for that. Okay, so in the last few minutes of my talk, I'd like to switch gears a little bit to talk about my third chapter of my thesis on gene flow and hybridization between the members of Culex pipiens, mosquitoes. As we saw in the previous chapters, gene flow and hybridization are common in this system. And importantly, gene flow changes ecology and behavior of mosquitoes in a significant way. So let's say in a northern big city, we have stereotypical above ground pipiens and below ground molestus. Pipiens likes to bite birds, molestus likes to bite humans. In some cities of the US, like New York City or Washington, DC, or Chicago, there are actual data suggesting that there's no negligible amount of gene flow from molestus to pipiens. And that changes behavior of above ground populations. Here's a piece of data from a study conducted in Washington, DC. What they found was that among the captured above ground individuals, those that had more molestus ancestry in the genome were more likely to bite humans compared to the other host-like birds. In other words, gene flow from below ground molestus might be driving the human biting in the above ground pipiens, which are otherwise bird biters mainly. This is particularly important in the context of disease transmission, as you can imagine, because the individuals of intermediate preference can be an effective bridge vector, transmitting diseases that circulate mainly in birds like West Nile virus into human populations. In fact, the hybrids have been implicated in the recent outbreaks of West Nile virus in New York City and Chicago. So for my chapter 4, I'm using these hybrid populations in the US to conduct a mapping study to identify which genes are underlying behavioral differences. But for my chapter 3, I will rather take advantage of the global samples collected for PipPop to investigate closely the pattern of hybridization in the genome across the wide range of populations. To do that, I'll have to introduce another closely related species of Culex pipiens, Culex quincofasciatus. Here I'm showing you a map of distribution of Culex pipiens in blue. That includes molestus as well, because there are ecotypes of one species, and Culex pipiens in yellow. So as you can see from the distribution, Culex quincofasciatus is global, tropical sibling of pipiens, and it has ecological and behavioral traits that are kind of mix of pipiens and molestus. So they mostly live in above ground environments. They can bite both birds and humans, depending on which populations you're looking at, I think. And they can mate in a small space. So it's like molestus in that sense. And they're active year-round as a tropical species. Importantly, pipiens and quincofasciatus extensively hybridize wherever they co-occur. So the green bands are all hybrid zones. Except for the regions that we have been talking about. So this region is really convenient in a sense, because we can ignore the quincofasciatus integration that complicates stuff further, which is interesting, but for the sake of study, it was really convenient, to be honest. So we think that these hybridization events across the globe might have created independent hybrid zones between pipiens and quincofasciatus, giving us exciting opportunities to investigate the genomics of hybridization. So I'm asking specific questions to understand how hybrids arise in this system, taking advantage of this natural experiments that created the independent hybrid clines in Culex pipiens. First, I'm asking how can genomes of two different species hybridize together, and how repeatable is the pattern across the independent admixture zones? To show you what genome data of hybrid zone look like, here's the PCA analysis using all the 777 individuals that have been sequenced for PipPop. And again, each dot indicates one individual. And first, I'm just coloring the Europe and North African individuals that we've seen already. And now you can see that pipiens molestus cline, and this is below ground, by the way, is on PC2 instead of PC1. And if you color North American individuals, it looks like this. So now you can see that they are totally different from European individuals, right? And they are actually spanning from pipmon to quincofasciatus. North American individuals are spanning across PC1, meaning that their genetic makeup is actually either pure pip, pure queen, or something in between, creating a continuous gradient of the ancestry. To illustrate further how the gradient is distributed geographically, here I'm showing you populations of West Coast, from Seattle to San Francisco to Fresno to San Diego. The y-axis are populations, and x-axis are the ancestry, either from pipiens or quincofasciatus. And on the right, if the dot is closer to the right, it is more like quincofasciatus, and if the dot is more like on the left, it looks more like pipiens in terms of genetics. So you can see this beautiful climb from north to south, from pure pipiens in Seattle to pure quincofasciatus in San Diego or Santa Barbara. So what's even more exciting to us is that with this whole genome data, we can further look at different parts of the genome, not just whole genome, and investigate the hybridization pattern across loci in the genome. I'm showing you here the allele frequency trajectories of 100 ancestry informative SNP loci across the genome. And ancestry informative loci mean that these loci are fixed for quincofasciatus allele at the southmost population, and pipiens allele in the northmost population. So they are like differentiated like according to the form at southern and northern edge of the populations. And you can see that these allele frequency trajectories are similar to the genomic ancestry trajectory that we saw at some times, but there are some loci that show steeper climb and others less steeper, like this, like a, almost like a line compared to the genomic climb. I summarize these trajectories using the width, and the center width is like this, and the center position of the curves like the panel on the right. And the width of the trajectories are particularly important because those with less steep slopes probably show the loci following isolation by distance, whereas those with steeper slopes like the edge may be the barrier loci in the genome, contributing to the segregation of important traits between pipiens and quincofasciatus. So right now we are investigating them further to see if there's any emerging pattern of the slope within the genome across different hybrid zones. So in summary, for this ongoing project, I'm taking advantage of independent hybridization zones to start asking how two divergent species hybridize and how the pattern of hybridization is repeatable across independent hybrid events. As the first example, I described the clear latitudinal hybrid zone in the West Coast, and I'm very excited to dig into further for other hybrid zones. Okay, so as a final summary for this talk, I have introduced the mysterious ecotypes of Culex pipiens that have fascinated scientists and public alike for decades, and our journey to develop them into a new model system of evolutionary genomics in the Anthropocene. So by the time, by now, I hope I have convinced you that this system can be studied to address some of the fundamental questions in ecology and evolutionary biology. And I hope that this talk fed your curiosity a little bit too. So thank you for listening. And I'll also have to thank a lot of people, Lindy McBride, Noah Rose, Molly Schumer, and Dina Fonseca for a lot of advice for my thesis throughout the 4 to 5 years of my journey. And the lab, including Tim here, who is taking picture, thank you. And research funding for sure. And all the people collaborators who tirelessly collect mosquitoes and analyze across the world. Okay, thank you. Yes.
All right. Okay, so I just came online for a second here so that I'll give a chance to those online And those that are not going to continue to talk, by the way, fantastic bagels and really good cream cheese here. So missing out if you're online, Zoe, I know you have an excuse. But those of you that want to ask questions right now, please go ahead as usual. Raise your hand so I can call on you. And then when you, well, if you're online, you can just say something. Those in here that want to talk, speak really clearly so that those online can hear. Okay, so we're going to go back to using Yuki's computer. Although, yeah, let's try that. So
Dina, I also made you a co-host.
Yeah, so
Yeah.
that people will ask first from the.
I think you can ask first.
Yeah, so very nice talk, fantastic.
Thank you.
Very, very cool data, and I can't wait to see what's coming out of it later.
Thank you.
And did you have any evidence of the Mediterranean mosquitoes, the molasses? They went probably they went north, and then do you have any evidence of after they diversify more the north areas, they would come back to Mediterranean and kind of make things noise here?
Yeah.
We don't have any noise, right? In our southern populations, right? Is that interesting? Is that surprising or?
Yeah. So, okay. The question was if the northern molluscus population we found was, is there a possibility that they are actually coming back to south and then make things complicated, especially the evolutionary testing complicated? And I think that is definitely happening to some extent. Maybe not like those Sweden below ground populations are coming back to Valencia or like Spain or something. But I believe that there is still ongoing gene flow to some extent between, especially nearby cities when people are moving back and forth. So, but if that is not like violating the overall pattern so much, both the amount of gene flow and the direction. If for example, gene flow is just nearby city, it's not like, from northern edge to southern edge continuously for decades, then I think our phylogenetic inference is going to be somewhat robust to that kind of scenario if we can look through across the genome. But I'm sure that that kind of like back migration is happening all the time.
So I have a follow-up on that because I think what you're saying is really biased by the fact that you're coming from a lab that works on Aedes mosquitoes. Because Culex mosquitoes don't travel like Aedes do. They don't survive drying eggs. So the age of Culex was when people carried big barrels of water and moved things, seriously. Now they don't move.
Right.
So I actually think that's part of the reason why you're having such a clean. I was asking, how did they? How are molestas moving across the world? I know that like in Russia, every time they would develop a underground thing, boom,
there it is. Yeah. I know. I mean, I don't know how exactly, but okay, my guess here is that they are transported not as eggs, but adults.
As adults?
As adults.
Oh, the story with the okay.
Yeah, so they're just like, so it's obviously speculative, but like those females that have mated without even blood meal can lay eggs in molestas species, which I didn't really talk about today, but it's actually one of the signifying property of molestas. So that's what we call autogeny, where the females do not have to take blood if they can consume nutrition from larval habitat. So they can lay first batch of eggs, survive, and then continue the population. And that's what I think that's one of the reasons why they can make it to the really closed manholes or like subway system where host is really scarce, right? So I think I agree that maybe unlike Aedes, the eggs cannot be transported like for hundreds of kilometers, but adults can.
Actually, that actually makes a lot of sense because it's molestas that got to Australia. They don't have pipiens. There's molestas that got to Argentina. They don't
Right.
have pipiens. So the far away and the ones that got here were already a mix. And that's me saying that. I'm like really curious to
There you
see
go.
what Yuki's going to find. But actually in your picture, you don't show, you said something about pipiens, but that picture that shows the Cuenca fasciatus pipiens and molestas, the straight line that you America, it never goes up all the way to pipiens. It stays in the middle.
You're talking
Back,
about
back,
this one?
Back. Yes, that one. Back one first, yeah.
This one,
So
yeah,
when you say that's American,
right,
it goes up all the way to pipiens, it
Yeah.
stays in the middle.
right. Yes, so that is also a really important point. So Dina was pointing out that the most pipiens like American pipiens above ground. So these are actually from like Seattle or Boston, those northern really part of America, has pipiens that do not look exactly like European pipiens, like pure pipiens, but they're kind of in between, like between pipiens and molestas. And that's what actually Dina was saying in the 2004 paper. But yeah, so we are rediscovering that for sure. And they can, for example, ecologically mate in a confined space, right? Is that so?
No, more confined than pure pipiens,
Right,
which doesn't,
which never really,
never mates.
yeah, mates in a confined space. So yeah, and yeah, molestas is the ones that are that only made it to Argentina. Pipiens never made it to Argentina, but molestas was recently introduced in Argentina and started to hybridize between quinks. So this line actually is coming from molestas to Cuenca fasciatus, right? And this line is more or less from Argentina. So individuals from Argentina are either molestas or Cuenca fasciatus or something in between, but you never see the pure pipiens in South America.
That was also true in Australia. That they didn't have pipiens either.
Yeah, but Australia is interesting because they don't have hybrids. They have molestas, Cuenca fasciatus, and that's it. Yeah.
So that's another interesting. Okay, I don't want to, this is my favorite topic in the world, so I should know. And questions from the audience. Please, if you're having shyness in terms of talking, they can't hear me.
Yeah, I think you
You
can unmute and speak a little bit, I guess.
Go ahead.
Does anyone have any questions from the Zoom?
Tell them to raise their hand. It's easiest.
Oh, yes. Zoe?
Yes, I have a couple of questions, but I'm also in the seminar class period. So if anyone else has any questions, like jump in. So one of them is I just went to AMCA and I was surprised that it seems like a very hot thing right now is using kind of like acoustic or optical tools to measure mosquito wing beat frequency and ID them that way. Like they have these massive machines that claim to be able to do that. I was wondering, after spending time with these species, would you think that there might be any difference perhaps in like mating swarms, et cetera, between the two species due to confined spaces?
Yeah, that's a great point. And I have no idea.
I don't think anyone's done it. I did a quick Google search. I was just curious about it. I didn't know if you'd I don't think you could probably notice it looking at them, but I didn't know if you had thought of it.
No, yeah, that's a great question. And there may be actually, if we precisely measure the wing beat frequencies between pipiens and molestas, like you said, the mating pattern or mating behavior itself is so different that there should be some differences, but my ears are not that good, I think.
Yeah, no. And those tools they look pretty expensive.
Yeah. Great question. Thank you.
It's actually past noon. Can you mute yourself again?
Sure. Yes. Give me one moment.
I don't want anybody to suffer.
Right. I don't see the. Oh,
You can just do. Oh yeah.
I think this one.
There you go.